This is podcast number 33. And we have been talking about cells all week. The last podcast talked about a specialized cell, neuron. This is another specialized cell, muscle cells. Muscles are excitable cells. They work with neurons, and most people are curious about muscles and how they work and how they respond to exercise, and we're going to talk about that today. There are different types of muscle cells. There are skeletal muscle cells. That's what we're going to talk about mostly today. And if you've heard about how they work, it's most likely you heard about skeletal muscle cells because that's some of the best studies. Cardiac muscle cells look similar in structure to skeletal muscle, but they're not quite as regularly organized, and they're smaller. Skeletal muscle cells are some of the biggest cells in the human body. And then there's smooth muscle cells that don't look like muscle cells like skeletal muscle at all, and that you don't see these stripes or striations in there, and they have very different energy requirements. But it's still muscle, it's still contractile. They are the things that line your gut. They are the things that line your blood vessels. If your blood vessels dilate or constrict, that happens with smooth muscle cells. All three types of muscle cells contract and get longer when they get shorter when they contract, get longer when they relax. They have similar mechanisms by how they contract. I'm going to show you a bunch of animations. Let's see if I can find them reasonably here. This one, I think. So these are as a movie of muscle cells contracting. These individual units of striations is called a sarcomere. They get closer together and further apart as they contract. Which muscle types grow bigger with repeated use? Cardiac muscle cells and skeletal muscle cells. One of the issues with heart disease is that if you put more strain on the heart by increasing resistance that the heart has to pump against, such as in hypertension, the heart will respond by getting bigger, and an enlarged heart is one of the problems in cardiovascular disease. And of course, you know that skeletal muscles respond to exercise with increased size. One thing that is important to know when you're looking at these pictures of muscle is that muscle cells are really big cells. It's very hard to see them unless you take a thin slice through the muscle cell, and it depends on how you slice it. So if we were going to slice through a donut or slice through a hand in what's called a longitudinal section, you get a profile like this. So if you slice from your wrist through your hand to your fingers, you get a profile of your hand. If you slice sideways through a donut, you get a profile of the donut. But if you're going to cut crossways across a hand or crossways through the donut, you get a different sort of picture. Like depending if you cut through the fingers, you get individual circle sort of profile, or an ellipse like this, depending on where you cut. So it depends on, it's important when you're looking at a micrograph of a muscle, how it was cut. Muscles are generally hot dog sort of shaped, long, tapered at the ends. And so if you cut through a hot dog like this, you might get a profile like this in a cross section. If you cut through the hot dog long ways, you would get in a long tube sort of shape. So this is a long ways cut through muscle that's stained with a stain that points out different features of the muscle. So these things here that are all over the place are nuclei, and you can see that there are lots of them. So muscle cells, as you are developing, start off as sort of eye-shaped not the letter I, but your eyeball sort of shape, in that if you're going to draw like a cat eye sort of thing. And with a single nucleus, and then as you develop, they fuse together and get much, much bigger. Muscle cells can be as big as like a millimeter in cross-sectional area, which is really big as far as cells go. And they can't, the nuclei come with them when these cells come together. So a mature skeletal muscle cell is going to have lots and lots of nuclei with it. There's a blood vessel through the muscle cell. These stripes or striations in here are called sarcomeres. We're going to zoom in on that. Here, this is an electron micrograph where you can see these very regular lines. This is characteristic of skeletal muscle in a electron micrograph. And these lines here come closer together during contraction, further apart when they are relaxed. And all of this, all the things that you can see in here are protein, except for this area here is membrane. If you cut the hot dog shaped muscle crosswise, this is the sort of thing that you see. And these are the ends of these protein filaments, the myosin and actin that we'll talk about. These are called the sliding filaments, and these are the things that slide past each other when your muscles contract. So these are sticking out at the end of the hot dog when you cut the hot dog in half, whereas in this, it's cut crosswise. Or sorry, longwise. Sorry about that. Okay, so this is a longwise view. Okay, and in a longwise view, you can see elements of these sorts of things on this picture. Okay, so you can see areas here where it looks thicker, and areas in here where it looks thinner, and then these ends, these dark bands on the end of the sarcomere. This is what we're going to talk about in this cartoon diagram. So the thick filaments here are the protein myosin. Myosin is a motor protein. And the thin filaments here are connected to other proteins on the end. And these are actin. So we've talked about actin before. Actin forms part of the cytoskeleton. Actin is all over the place in all kinds of cells, not just in muscle cells, but in muscle cells it's arranged into these thin filaments that are next to these thick filaments. And what you're going to see is these little bumps hanging off of these thick filaments are going to walk along these thin filaments, and they're going to pull on them. And they're going to pull toward this end and pull toward this end. So if these guys are pulling this way, and then these guys are pulling this way, what's going to happen is these two bars are going to get closer together. So when it relaxes, the two bars get further apart, and the myosin heads are not pulling on the actin. When it contracts, they are pulling on the actin, and the actin filaments get closer together, and the two bars get closer together. Okay, so the sarcomere length is from one bar to another. This would be fully contracted. You can see that they can't really get any closer together than that. And this would be fully relaxed in that they can't really get further apart than that. Okay, actin pulls toward the center, start uncontracted to contracted. Okay, so we're going to see this myosin head in an animation here walking on that actin filament, and it's walking toward this end, pulling this end toward the middle, and this one is pulling this end toward the middle. So it's like both of these are tug-of-war teams back to back, and both of them are pulling toward the middle, and they're pulling against this team here and this team here, so the ends come closer together. Okay. This is just a myosin monomer, so a bunch of these are wound together to give this thick filament, and it's just looking at one individual filament and head in this, unwound together. So the myosin thick filament is in the middle. It stays in place and it pulls on the actin relative to the myosin. If you put a bunch of them in a diagram, you'd see one filament here, and then all of them twisted up together into an actin or myosin filament in various shades of yellow and red here, and in blue that's the actin that it's going to bind to. Okay, in cross section there would be these individual heads sticking out. So in this way they're laying side by side. If you in the hot dog here, and if you turn the hot dog 90 degrees and you cut it, you would see the ends of the myosin sticking out here and the ends of the actin sticking out there. Okay, we're going to watch this little movie here about how myosin walks on actin. I think this is it. Yep.
Muscle myosin is a dimer with two identical motor heads that act independently. Each myosin head has a catalytic core and an attached lever arm. A coiled coil rod ties the two heads together and tethers them to the thick filament seen on top. The helical actin filament is shown at the bottom. In the beginning of the movie, the myosin heads contain bound ADP and phosphate and have weak affinity for actin.
Weak affinity means it binds actin weakly.
Once one of the heads docks properly onto an actin subunit, phosphate is released.
This is phosphate that comes from ATP. ATP, you're going to see, is a really common energy molecule in cells. We saw it before when we talked about how the sodium-potassium ATPase works, and we're seeing it here in how myosin works.
Phosphate release strengthens the binding of the myosin head to actin, and also triggers the force-generating power stroke that moves the actin filament. ADP then dissociates, and ATP binds to the empty nucleotide binding site, causing the myosin head to detach from the actin filament.
So this is an important point and sort of a cool feature of muscle, in that you normally think of muscles contracting need lots of ATP, and in a sense that's true, but strictly speaking it's not true. So ATP, as you just saw there, has myosin unbinding from actin, which is associated with relaxation. So it happens a bunch of times when it's cycling and contracting, but really you need ATP for muscles to relax. One of the things that happens when an animal dies is that they get very stiff. This is called rigor mortis. It's because their cells are no longer producing ATP, and their muscles are stuck in the bound state, and they need ATP to be added in order to break that bond and for their muscles to appear loose. So when you can't make ATP anymore, you go through rigor mortis, and then eventually what happens is these proteins break down as the body decomposes, and the corpse gets more loose. But rigor mortis is caused by no more ATP, and when the actin is bound to the myosin in the muscle is stiff.
On the detached head, ATP is hydrolyzed, which recocks the lever arm back to its pre-stroke state. Thus, like a spring, the arm stores the energy released by ATP hydrolysis, and the cycle can repeat. The actin filament does not slide back after being released by the motor head, because there are many other myosin molecules also attached to it, holding it under tension.
So what you'll see is this is really slow speed, but in a real contraction, like when you lift a barbell or something like that, it's going to go through this cycling mechanism several times, hundreds and hundreds of times as it contracts. And then You'll have an oppositional muscle, like your triceps, for example, pulling and physically bringing back the muscle to the relaxed state. Okay. Another aspect of this system is calcium. And calcium is released into the muscle to allow the contraction to happen. And what calcium does is gives myosin a place to bind on actin. So in that last diagram, they didn't show you this, just to make it simpler, but there's another protein that winds around the actin filament called tropomyosin, and then some calcium binding proteins called troponin. When there is calcium in the muscle, these calcium binding proteins, troponin and tropomyosin, bind calcium, change shape, and then move out of the way so that myosin can walk on actin. But if there's no calcium around, they move back and they cover up the spots for myosin to bind actin. So if there's no calcium inside the muscle of the cell, it is in the relaxed state. And only when calcium is in there can muscles contract. Okay, so that's important to keep in mind as we go forward. Okay, the myosin binding site gets blocked by tropomyosin and is only moved out of the way if calcium is around. All right, so how does calcium get there? All right. In skeletal muscle cell, again, these are giant cells, and they are formed by lots of little cells fusing together, lots of nuclei in those little cells. When those cells fuse together, there's one giant membrane that covers the whole thing, called the cell membrane or plasma membrane. And if the muscle cell was relying on diffusion alone to cause the muscle cell to contract, it would happen really slowly. So there's evolved this mechanism by which a change in membrane potential, which happens very fast because in fact it essentially travels at the speed of electricity, can be transmitted into the belly of the muscle. So even though this is a very large cell, up to a millimeter thick, there are these invaginations of the plasma membranes called T-tubules. So the T-tubule, it's like the plasma membrane is like a balloon, and in the surface of the balloon, you stuck your finger into the balloon, poking into the middle of that hot dog, and your finger is still covered by the balloon. You haven't popped the balloon, but now you've got a invagination, a hole that is going into the belly of that hot dog. And because it's part of the plasma membrane, if the plasma membrane depolarizes, then this part also depolarizes because it's still part of the plasma membrane. And this part is connected to the middle of the cell. Okay? So it's like a spy or a tunnel into the middle of the cell that's connected with the outside of the cell. This wasn't discovered until the 80s. They knew that muscle cells were stimulated and then there was calcium release, but they didn't know how that happened. And in the 80s, it was discovered that there's this connection between the outside of the cell, which is connected to this membrane, which goes all the way into the belly of the cell, called the T-tubule, and the middle of the cell. And in the middle of the cell, you have the endoplasmic reticulum is modified in muscle. Now it's called the sarcoplasmic reticulum. It stores all kinds of calcium. And what you're going to see is going to happen is that as this cell depolarizes because an action potential has caused it to depolarize, that depolarization travels really quickly down into the belly of inside the cell, and then there's a protein bridge between the outside and the inside of the cell. A protein on the outside of the cell reacts to that change in voltage, is connected to a protein on the inside of the cell, that reacts to that change in voltage, and that protein releases calcium when it changes shape. Okay, so action potential enters the muscle interior through these T-tubules. That happens really quickly because that happens at the speed of electricity. The electricity has changed the membrane potential, and the membrane goes all the way into the middle of the cell. The T-tubules are tunnels branching from the plasma membrane. They're connected to the plasma membrane. They're part of the plasma membrane. They conduct the action potential to the middle of the muscle, and then they are physically connected by protein bridge to the inside of the cell, an organelle on the inside of the cell called the sarcoplasmic reticulum that contains a bunch of calcium. It's basically a big bag of calcium. And then when it gets that signal from the outside of the cell, it releases the calcium. The calcium binds to the troponin. The troponin moves out of the way, and now muscles can contract. Okay, calcium floods in. Okay, here's what sarcoplasmic reticulum looks like in a longitudinal section in an electron micrograph. Okay, and we're going to now put it all together in another animation. Maybe? Yeah, this one. This one.
Muscle cell. An action potential sweeps over the plasma membrane of the muscle cell. The action potential releases internal stores of calcium that flow through the muscle cell and trigger a contraction. Muscle cells have an elaborate architecture that allows them to distribute calcium ions quickly throughout the cytosol. Deep tubular invaginations of the plasma membrane, called T-tubules, crisscross the cell. When the cell is stimulated, a wave of depolarization, that is, an action potential, spreads from the synapse over the plasma membrane and via the T-tubules deep into the cell. A voltage-sensitive protein in these membranes opens a calcium release channel in the adjacent sarcoplasmic reticulum, which is the major calcium store in muscle cells.
So here's that protein bridge I was telling you about. Here's the outside of the cell connected to the T-tubule. Here's the inside of the cell connected to the sarcoplasmic reticulum. And here's the protein bridge. This is going to change shape when it gets the signal. That causes this to change shape when it gets the signal, and calcium comes out. This is the thing that reacts to caffeine that I told you about in another podcast. So when you drink caffeine, this is open more often than normally without caffeine, and it can make your muscles have these little microcontractions and give you the shakes.
Thereby releasing a burst of calcium ions all throughout the cytosol of the cell. Within a contractile bundle of a muscle cell, called a myofibril, the calcium interacts with protein filaments to trigger contraction.
So what they mean is calcium is going to bind to those troponin and tropomyosin proteins, so that tropomyosin gets out of the way on the actin and allows these myosin thick filaments to walk on the actin and pull the sarcomeres closer together.
Contracting unit, or sarcomere, thin actin and thick myosin filaments are juxtaposed, but cannot interact in the absence of calcium. This is because myosin binding sites on the actin filaments are all covered by a rod-shaped protein called tropomyosin. A calcium-sensitive complex, called troponin, is attached to the end of each tropomyosin molecule. When calcium floods the cell, troponin binds to it, moving tropomyosin off the myosin binding sites. Opening the myosin binding site on the actin filaments allows the myosin motors to crawl along the actin, resulting in a contraction of the muscle fiber. Calcium is then quickly returned to the sarcoplasmic reticulum by the action of a calcium pump.
Okay, that calcium pump is called SERCA, and it returns calcium into that bag called the sarcoplasmic reticulum, and that's what causes the tropomyosin to move back over the site where myosin is going to bind, and that's what allows the muscles to contract. So again, that requires ATP. And so again, the relaxation part of a muscle contraction relaxation cycle is the part that requires energy. Just like you saw that you have to add ATP to get the myosin head to come off or relax from actin, you have to add ATP in order to pump the calcium out of the system. So again, relaxation is the thing that requires ATP, not contraction.
Without calcium, myosin releases actin, and the filaments slide back to their original positions.
Okay, let's finish this thing up here. All right, what happens if you work out? So here's an experiment that has been done with rats, and they compared what happens to a mouse in its left leg, where you remove the gastrocnemius, which is the big muscle at the back of your lower leg, and kept the plantaris, which is another muscle which is much smaller, but does a similar sort of action on the leg, so mice or the rats can still walk. And on the other leg, they kept everything the same, and so they compared the size of the plantaris on the leg where it had to do more work because the gastrocnemius was missing over here, to the other leg where it had to do less work because the gastrocnemius is right there. Okay, so over time, okay, this is when they 5 days after they did the surgery, 30 days after the surgery, and then 60 days after the surgery, you can see that the plantaris muscle on the side that had the gastrocnemius removed got lots bigger, and the other muscle also got bigger. Why did it also get bigger? Because these mice are growing. These rats rather are growing. These are young rats, and they're growing, and the muscles are going to get bigger anyway. But the one that had to do more work grew lots faster. Well, did it grow faster because they got more muscle cells, or did it grow faster because they got more muscle protein? Well, they also looked at the amount of DNA in the different cells under those two different conditions. Okay, and you can see the amount of DNA goes up, but it doesn't go up quite as fast, and it doesn't go up very much at all in the control side, where they did not remove the muscle. So the reason it does go up is because new precursor cells, stem cells, are fusing to the existing cell, making that cell bigger and adding their nucleus to that cell. Again, these are multinucleated cells formed from fusing of smaller cells. But it's not like the original cell became 2 cells or became 4 cells. That's not what happens. So there's a saying in muscle biology that muscles hypertrophy in vertebrates, but they don't go through hyperplasia. So hypertrophy means they get bigger with exercise. Hyperplasia means more cells. So hyperplasia is what happens in cancer, for example. You just keep on going through mitosis, so you get more and more cells. In the case of skeletal muscle in vertebrates, things with backbones, under conditions of exercise, the muscle cells get bigger, but they don't get more muscle cells. So if you look at a weightlifter versus a world-class marathoner, the weightlifter is going to look like they have much more muscle mass, and they do, but they actually have the same relative number of muscle cells. Every individual is going to have slightly different number of cells, but same relative number of cells in their muscles, comparing a weightlifter and a marathon runner. Okay. Seems like there was one more thing I wanted to tell you about that. Oh, yes. Sorry, and a little stumble there. So why do they get bigger? They get bigger because all that stuff that we saw about, the actin and the myosin, in order to do more work, to lift more, because that gastrocnemius muscle was missing, you need more contractile units, so more actin and more myosin. And so when someone goes through a weightlifting program and they get bigger by virtue of making more actin and myosin protein. And the individual cells get bigger, but they don't get more cells. So hypertrophy, not hyperplasia.